Hello, it's Tam here and welcome to today's lesson. So I'd like you just to pause the video and have a go at this question here, please. Okay, so let's have a look at the answers. There are two main roles of DNA, and the first one is DNA replication. Now, DNA replication, you hopefully remember from last week, is really important to make sure during cell division that you are replicating the DNA for the daughter cells that are produced. And the second role of DNA, this is actually what we're going to be looking at today, and this is protein synthesis. You might want to take a second just to loll at this cartoon down here. Okay, next I'd like you to pause the video and have a go at this activity. Give yourself 5 minutes. Use the diagram to explain how DNA codes for proteins. You've got no limit of marks, and we're going to return to this at the end of the lesson. So pause the video now. And your learning objectives for today are these. So if you could, again, pause the video and I'd like you to jot down your confidence ratings on your progress bar. Okay, next, can you please grab a mini whiteboard and have a go at these couple of questions. So why is the code called a triplet code, and explain how genes code for amino acids. And you might want to use this diagram here just to help you a little bit. So pause the video. Okay, let's have a look at the answers then. So the code, now when we're talking about the code, we're basically talking about the DNA sequence. So the sequence of bases on the DNA molecule. And it's called a triplet code because you need 3 bases to code for 1 amino acid. Okay, now we can actually kind of see that in this diagram below, and this actually helps us when we're explaining how genes code for amino acids. And what we can see here in this diagram is the RNA sequences. So on DNA, it's called a triplet code. On RNA, it's actually called a codon or anticodon, depending on whether you're looking at messenger RNA or tRNA. And this table basically shows how RNA can be read in 3 base sequences to code for each amino acid. So, for example, we can see that this 3 base sequence on RNA, uracil, 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 codes for this particular amino acid. Okay, and you can actually follow the directions around the side of this image. So we can look for the first letter of the sequence, for example, if it was A, then the second letter, maybe the second letter was C, and that would be this column. And then maybe the third letter was G, so ACG. We can see that this 3 base sequence codes for this amino acid here. So this is why it's called a triplet code on DNA. And we're going to look a little bit more about explaining this process of how genes code for amino acids. Okay, and this is a little overview of protein synthesis. So there are 2 key parts of protein synthesis. The first part is transcription, and that, as you can see, takes place in the nucleus, so that's here. And the second part is translation, and that takes place outside in the cytoplasm on ribosomes. Okay, and these ribosomes can actually also be located on the rough endoplasmic reticulum. Okay, and you can kind of see a ribosome here in grey. So the first one, transcription. Now, as you guys will remember from GCSE, you have lengths of sections of your DNA called genes, and genes, as we know, are used to code for polypeptide chains that will then fold into proteins. Okay, so what we can see here at this point is here's a gene, and the DNA has unzipped itself, and we've made a little copy of that gene called a messenger RNA molecule. And we can see that this is an RNA molecule because it's single-stranded, if you remember back to when we looked at nucleotides. And this messenger RNA molecule, or copy of that gene, is going to move out of the nucleus via this little hole or a pore, nuclear pore, and it's going to move to a ribosome. And then on the ribosome is where the second stage of this process takes place, which is translation. So in translation, we're going to look at this code sequence on the messenger RNA, and we're going to form a polypeptide chain. And what you can see here is that we're going to use little molecules called tRNA or transfer RNA to bring specific a specific amino acid each time to start forming a polypeptide chain. Okay, so for example, this tRNA molecule here will only ever carry 1 specific amino acid. Okay, and the tRNA 3 bases at the bottom that you can see here will be complementary to the 3 base sequence on the messenger RNA, and we're going to come back to those names in a minute. So here's a little bit more of a detailed diagram of that. So again, here on your DNA molecule, you can see we've got sections of that DNA called genes. Okay, so here's in dark blue is gene 1, in yellow is gene 2, and then in green is gene 3. Okay, so maybe this gene here, for example, gene 1, maybe that codes for a protein that is used in digestion, some sort of carbohydrates. Maybe gene 2 codes for a protein that forms a channel protein in the membrane. So you can see that this is why, you know, DNA codes for certain characteristics and traits, is that it makes proteins that are able to do certain jobs. Okay, so we can see if we look at the DNA strand, and here we're looking at the template strand. So you know DNA is a double helix, and one of those strands is used as the template strand for forming a messenger RNA molecule. Okay, and so ultimately what's going to happen is when the DNA unravels, as we saw over here at the top, the template strand is now sort of only got one side of the double helix, if you like, and so these bases here are now exposed, and you can have little messenger little RNA nucleotides that come along and form complementary base pairs with that DNA molecule, and then they get bonded together to form a messenger RNA strand, and that is what you can see here in red. So what we'll notice is the 3-base sequence on DNA. So here on the DNA, this 3-base sequence is called a triplet, and you can see it's complementary to a 3-base sequence on messenger RNA, which is called a codon. Okay, and you can see that here, there's the name codon. And then the tRNA molecules that you can see over here in this diagram, they bring one specific amino acid, and they have anticodons that are complementary to the messenger RNA codon. So this stage here, translation, happens on the ribosome, and the transfer RNA would bring along a specific amino acid, and we can see this codon is going to ultimately correspond to an amino acid here. And then we can bond those amino acids together, and you can see we're actually forming a polypeptide chain down here. Okay, so the 3-base sequences have different names depending on what molecule you're talking about. So it's really important that you use the right name for the right molecule. So if you're talking about the 3-base sequence on DNA that codes for a specific amino acid, you would be talking about a triplet or a triplet code. If it's a 3-base sequence on messenger RNA, it's called a codon, and if it's on tRNA, it's called an anticodon. So this is quite useful because if someone's telling you about the process of transcription translation, you can just listen in and check which molecule they're talking about by listening to what one of those 3 key phrases they use. And down here we've got another diagram. So again, we know that ultimately DNA codes for protein synthesis, as we said earlier, and we can see that we've got these stages that we've spoken about before. Next, please can you pause the video and go and watch this video that's linked in your activity. And just as you're watching the process of protein synthesis, I'd like you to jot down the keywords of that process and make a record of those. We're going to use those later for writing descriptions. Okay, next, I'd like you to have a go at the transcription activity. So for this, please can you individually make a list of all of your keywords that be involved in the process of transcription. So you did make a list of those a minute ago when you watched that video, but try and do it from memory, okay? Then you might want to check on your list, see if you missed any off. And next, I'd like you to use this diagram here and your knowledge to write a bullet point explanation. So pause the video, spend 2 minutes listing your keywords, and then spend a further 4 minutes writing your bullet point description. Okay, so let's take a look at the answers. So you might want to just pause the video and check that you've got a similar description. It doesn't have to be identical, but what you should have is these keywords in bold used in the correct order, okay, and the correct context. Just a couple of things to remind you about. Do just remember to spell complementary with an E, not an I. And also the nucleotides, the RNA nucleotides, they're forming complementary base pairs with the DNA template strand. These are called free RNA nucleotides, meaning they're not bonded by a phosphodiester bond to another RNA nucleotide. Okay. So next, I'd like you to go back to your progression line and just to see if your confidence has changed for any of these 3 learning objectives. Okay, and for the next activity, I'd like you to spend 30 minutes and to complete at least 2 of these. So if you're not very sure about this whole process, you might want to try activity 1. This is just a basic close exercise, so just to go through the process. There's also that video that you can watch again. If you feel okay and you want to sort of challenge yourself a bit, then have a go at activity 2. This is like recall questions, and then question 10 is a bit of a summary sort of application question. And then if you feel really confident, then you can have a go at activity 3 and just research your own question about protein synthesis. Or you might want to use some of the questions here. And then do share your answers with your channel on Teams. Okay, so pause the video, 30 minutes. Okay, finally, I'd like to just talk to you about the process of tRNA activation. So as we know, tRNA is a polynucleotide, which means that it has many nucleotides. So poly means many, nucleotide is the unit. So nucleotides we know make up DNA and RNA. So here we can see we've got many nucleotides bonded together to form this larger molecule. And then we can see at the bottom here, we've got a little section where basically we're going to bind an amino acid, okay? And when we bind an amino acid onto this tRNA molecule, this is what we call tRNA activation. Now, this is actually going to require energy. Another thing just to note is that you've got down here the anticodon loop. So this is where you'd find the 3-base sequence that is the anticodon. Okay, so that's what would form complementary pairings with the codon on the messenger RNA. Okay, so here we can see we've got our tRNA molecule, transfer RNA molecule, and here is the anticodon AAA, adenine on that tRNA molecule, and we're going to add this specific amino acid. So just a top tip here is whenever you're talking about tRNA moving or transferring amino acids, they only carry one specific amino acid each time. So whenever you're talking about tRNA, please make sure that you say each tRNA carries one specific amino acid rather than tRNA carries amino acids, because that implies that they carry more than one, but each tRNA will only ever carry one specific amino acid. So let's look at how we bond this amino acid onto this tRNA molecule. We are going to use a little bit of energy. And you'll remember that the energy packet that we use is called ATP, and that when that's broken down into ADP and phosphate inorganic, that it releases a bit of energy. And that energy is used to activate this tRNA molecule by attaching this amino acid, this specific amino acid, onto it. So here we've got our activated tRNA molecule with its specific amino acid, and that's tRNA activation. So finally, can you go back to your original description that you did from this diagram at the very start of the lesson? I'd like you just to see if you can add any extra detail into it. Okay, so pause the video, take a few minutes to have a go at that, and then come back. And finally, please could you go back to your progression line and just re-rate your confidence for these 3 learning objectives. Have a lovely day. Bye.